to this departmental seminar of the Department of Pharmacobiology and the Center for Research in Aging. So today we have a very special guest, Professor Glenn Milhauser from the University of California of Santa Cruz. For those of you who just joined today, we have been here and in Zacatenco in a 3-day workshop on EPR spectroscopy to study metals in biology. And we thought that as part of our workshop, we will have Glenn give this departmental seminar. He's an expert on prion protein and its interactions with metal ions like copper, which is also our favorite metal. And I should say, he's here thanks to the support of UC Alianza MX and Simbe staff. So the University of California has this Alianza MX office, and their main purpose is to strengthen relationships and academic exchange with Mexico. And so Simba staff, we are very lucky at Simba staff that thanks to the efforts of our administration, we have this agreement between the University of California and Simba staff to fund collaborative projects. And we've been very lucky to be funded with one of these grants in which Glenn participates. And we also had here Dave Britt from UC Davis. He had to leave today for another trip. But so this is made possible all to this collaboration between the University of California and Simba staff. And so for those of you who don't know Glenn yet, because you just joined today, so Glenn, he did his bachelor's degree at the California State University in Los Angeles, the city where he grew up. So he kind of feels at home in Mexico City because it's also a huge city. Then he did his PhD in physical chemistry at Cornell University. And then also at Cornell University, he moved to the Department of Pharmacology to do his postdoctoral training. And he's been for a while now professor at the University of California at Santa Cruz, where he is distinguished professor. And he's also department chair of this Department of Chemistry and Biochemistry. And we're very happy to have Glenn here. I have to say I have followed his work for probably 2 decades now, since I was a graduate student, looking at when he first started studying copper binding to the prion protein. And so now we're very happy to finally meet in person and be collaborating with him in some projects. And thank you, Glenn, for being here. Oh, okay, okay. Okay.
While Liliana's doing this,
sorry.
We've actually published a paper together, and this is our first time meeting in person, and it's been a wonderful workshop. So Macintoshes are always so protective and secure, and it asked me like 5 times, "Are you sure? Are you sure? Change your system settings." And so I said, "I am sure," and I changed my system settings. Now, are you sure? Do you trust Liliana to do this? Oh, it's doing it again. Oh, wait, we got that. See, I turned it on. Oh, there it is. Okay. Now I can find the talk. Okay, got it. And I'll open my Simba stuff. Now I have to find the talk. Okay. And then, oh, can you do that setting? Yeah. You have to show me how that's done.
So you go, I think you go to view and center to screen.
Okay.
And then it works.
Great. Okay. Oh, and it's started. It was at the end. All right. Let's start there. Play. And then view. Enter full screen. All right. We're there. Okay. Well, thank you. Okay, now let me make sure I have everything. So the microphone, advance the screen, and then this is a pointer. And I know it has a button. Oh, We're set. Okay. Well, first of all, thank you for the lovely invitation. It's a pleasure to be here. I haven't been to Mexico City before, but we're having a great time, and touring around, and the science and everything is just absolutely wonderful. So thank you all for coming, and again, Liliana, for the lovely introduction. So before I start, let me mention the folks that I've worked with. The work I'm going to tell you about today is highly collaborative. We are chemists, biochemists, depending on how you want to characterize as biophysical chemists. We collaborate with folks that help us enhance, let's see, oh, there we go. Dave Britt, who was part of the meeting and ended up leaving at 5 o'clock this morning, poor guy, and his former, yeah, his former postdoc, Lizzie Tao. Also, I'll be showing you some electrophysiology, and this is all done with our collaborator, David Harris. We've collaborated for quite a while now. We're on each other's grants, and we really have a nice interaction. And the work I'll be showing you is from his postdoc, Bei Wu. So let's get into some of the background. And the organization of what I'm going to talk about today is, first of all, I'll give you some background on prion diseases. And I know some of you, especially in Liliana's lab and so forth, are experts in this. But let's go through some of that. And I'm going to talk about a very curious phenomenon that we call deletion disease. And I'll explain what I mean by this. But it sort of changes the way one views neurodegenerative disease. We always think of it in terms of aggregation, and it's not necessarily the case. I'll introduce the electrophysiology techniques that we use. We'll talk about PRP structure. I know this is more of a biological audience, so I'll explain how we use various magnetic resonance techniques. And then some what I consider rather surprising work lately is from glycosylation. As many of you know, proteins in general can be highly glycosylated, and we've wondered the effects of glycosylation on the prion protein. So I believe Jan and Carolina were talking about this morning in the student presentations or postdoc presentations about the prevalence of Alzheimer's disease. These are some data from the United States, and as you folks know, Alzheimer's disease is increasing as our society gets older, as we have more people with gray hair. The incidence of these diseases is becoming much more prevalent. 6th leading cause of death, and 1/3 of the individuals over the age of 80 show some degree of dementia, and in most cases that dementia, or many cases, is Alzheimer's disease. The cost that goes into this, and these are US numbers, are, you're looking at $250 billion in medical care per year. But in addition, and we're actually seeing this, I mean, my father-in-law, my wife's father, is now starting to suffer from dementia. And the amount of care that it takes from the family, and now that his wife can't work because she has to be at home with him, the amount of care that my wife is putting into this, and this all pulls people out of the normal workforce. And so there's these sort of undocumented, if you will, ways of that it costs our society, throughout the world in terms of these neurodegenerative diseases. And of course, these costs are not going down for many reasons, and this is projected to be $1 trillion per year by the year 2050. And I would guess that it's actually going to be quite a bit more than that. So prion diseases are really interesting for a number of reasons. So first of all, I'll show you what the prion protein looks like. Unlike Alzheimer's disease, which I won't spend too much time talking about, but Alzheimer's disease, as many of you know, is initiated by a fairly small peptide. It's a proteolytic product of what's called the APP, the amyloid precursor protein. It doesn't have much structure on its own. And this is the case for several different types of neurodegenerative diseases. But the prion protein responsible for these diseases is a complicated protein. It has multiple domains. It's glycosylated. It's a metalloprotein, and so forth. So in and of itself, it's an interesting protein. But also what makes it interesting is that as opposed to being just a human disease like Alzheimer's disease, this is a disease that affects a number of different mammalian species. And you folks probably have heard of some of these, but there's scrapie in goats and sheep. Chronic wasting disease is a huge problem in the US, in the Rocky Mountain states. This disease is spreading like crazy, unabated, among deer and elk in the Rocky Mountain states. And it continues all the way up through Canada. The agricultural departments will go in, they'll kill off the animals that are showing diseases, they'll separate the carcasses from the healthy animals, and the disease will continue to spread. And so there's really no stop to this, and literally we could see the end of certain deer and elk species because of this. There's, of course, the most well-known societally is mad cow disease, and there's enormous effort that goes into slaughterhouses to make sure that these kinds of tissues aren't propagated into the human food chain. There's Kuru, which came about from ritualistic cannibalism. This was fairly prevalent in the Foray people of tribal New Guinea back in the 1950s, 1960s, and it was Carlton Gajdusek and ultimately Stan Prusner who showed that this was an infectious disease that came about from cannibalism. They no longer practice that, and fortunately, Kuru is now all but disappeared. But it's a morbid but very, very interesting story. And then in modern society, in our societies, there's Creutzfeldt-Jakob disease, which is similar, has some similarities to Alzheimer's disease. It's thought of as being, and we were discussing this before the talk, it's thought of as being exceedingly rare, human prion disease, but in fact, they're not as rare as one might think. And I've actually known of several cases where individuals passed on from various forms of human prion disease. About 1 in 6,000 deaths in the US are from prion disease. Creutzfeldt-Jakob disease, you can almost think of it as Alzheimer's disease on steroids. It goes very, very fast. People can go from initial symptoms where Alzheimer's can take a decade to develop. Creutzfeldt-Jakob disease, which is the most common of the human prion diseases, it can go from diagnosis from the first symptoms to death in about 2 years. In addition, there are not just cognitive consequences, there's motor neuron consequences. People lose coordination. It affects, effects are throughout the brain, cerebellum and so forth. And so people have all sorts of physiological difficulties. So this now touches on what I was saying before. We tend to think of neurodegenerative diseases as being diseases of aggregation. And in most cases they are, with, a matter of fact, in the previous, session, Jan was talking about the aggregation of A beta and so forth and the extracellular plaques, and that's absolutely true. And so there's this mindset among those of us, many of us in the neurodegenerative field, that all the neurodegenerative diseases arise from aggregation. So you have Alzheimer's disease, you have extracellular aggregation of A beta, you have intracellular aggregation of tau. In Parkinson's disease, you have aggregation of alpha-synuclein. In the prion disease, you have aggregation of the prion protein. And typically, these aggregates form these beta sheet-rich structures like you see over here. And when those form, this is a plaque out of what VCJD is actually derived. This is post-mortem from an individual who suffered Creutzfeldt-Jakob disease, but initiated by consuming mad cow tissue. And this had happened to a number of individuals in the United Kingdom back in the 1990s, I believe it was. But anyhow, there's these plaques in there that are very beta sheet rich. You can actually grow these things in the laboratory, and they'll form these kinds of fibrils, and people in the neurodegenerative field are very, very familiar with what these fibrils look like. But it's not always the case. And so before I get into some more details, let me show you the architecture of the prion protein and explain what makes it more complicated and a little bit more interesting. And I'll say right now, this is not actually what it looks like. I'm going to show you data that casts doubt on the disposition of these different domains, and I'll explain in a few minutes. But just looking at this in terms of sequence space, so first of all, it's about 210 amino acids, so it's not a very, very large protein. It's a membrane-associated protein. It has a GPI anchor down here, a glycophosphatidylminoxidil anchor that has a lipidic tail on it, so that interdigitates into the outer leaflet of membranes. It has this helical C-terminal domain over here. It's doubly glycosylated typically. Actually, you can find different glycoforms. You can find it monoglycosylated, diglycosylated, and sometimes unglycosylated. And then it has this extended N-terminal domain, about 100 amino acids. And within this domain, there are these two sort of what we refer to as polybasic segments. These are segments that have basic amino acids, arginine and lysine, and so they're positively charged. And this will play a role in a few minutes, as I'll show you, in terms of the toxicity of the protein. And then there's this very, very interesting, as those of us in the field know, this octarepeat domain. And this domain is a repeat. It's a really, really simple repeating sequence, PHGGGWGQ, and typically 4 times. And just to say, all of us have the prion protein, and the central nervous system is rich with this protein. It decorates the surface of nerve cells. It's found in synapses, presynaptic, postsynaptic, and right. And so anyhow, you have typically, most of us statistically will have just 4 repeats. However, there are certain family lines that have expansions of these repeats, up to 5, 6, 10, 15, repeating segments. Those individuals, curiously, are very, very prone to develop different inherited forms of prion disease. So anyhow, so we'll pay attention to that. And then there's this little hydrophobic region in here, which will play a role in a couple of minutes. So anyhow, this is in physical space, you know, in molecular space, and this is in sequence space up here. So those are the different features of the prion protein. Now, what I want to talk about, and this is what I was referring to as deletion disease. So this is a very, very simple schematic here. And what you've got is, I'm just showing, so a mouse, you know, mouse model, prion protein, all the amino acids. And if you create a transgenic mouse where you've deleted the N-terminal domain, that mouse is absolutely fine, okay, no problem whatsoever. If you delete the entire protein, okay, that mouse, for the most part, is fine. Not totally, but it will survive, okay? It will show some, not quite pathology, but histology that's not compatible with a properly developed animal in terms of myelination and, you know, nerve size distribution and so forth, but it will survive. However, so this is my very, very simple graphic. Okay, watch. Okay, so if you pluck out a few amino acids, about 20 amino acids, out of the middle of this protein, it is absolutely lethal, okay? So how can this be? And if we think about the human genome, we code for, you know, depending on the different polymorphs and so forth, 10 to 20,000 different types of proteins. And here we've got this really, really simple protein, and you delete 20 amino acids out of the middle, and it is absolutely toxic, okay? How can this be? So I'm going to show you a video. Now, this is not our data. This is fairly old data, but it's from Adriano Aguzzi's lab in Switzerland. He's one of the lead prion researchers. And this is an animal, and you will see the phenotype that's associated with missing those amino acids out of the center of the protein. So I'm going to just run this little video, and you'll see what happens. So this is a very young mouse, and it is showing basically the symptoms of prion disease. So it loses coordination, it falls over, you know, it's very sad, okay? But if you were to take a mouse that has its normal prion protein and you were to inject it in the central nervous system, in the brain, with prion protein aggregates, you would see a very similar phenotype. Also, this animal, when it's dissected post-mortem, you look at the brain tissue, there are no prion aggregates. There's no aggregation whatsoever, okay? So what you've got is a disease where the aggregation is forming a disease, but you can replicate the disease by just knocking a handful of amino acids out of the middle of the prion protein, okay? So what this tells us is that the disease can't, you know, it's very possible that the aggregation is almost a distraction from what's really going on, okay? There's something about the monomeric form of the protein, okay, that is allowing this neurodegenerative phenotype to develop. Okay, so now, where is that section? Okay, that section that was knocked out, the 20 amino acids. Well, it's this little yellow segment right here, and we call it, very imaginatively, we call it the central region, okay? And it's central because it's between the N-terminus and the C-terminus, okay? And that's where that is. So now what we want to do is explain how this is happening. So this is now David Harris's work. And as I say, we collaborate with him. And as many of you know, working with transgenic animals is informative but incredibly expensive. And you'd like to have a faster read-through to figure out what's going on. And he showed many years ago that we can use whole cell patch clamp and look at transmembrane currents. And so what you're seeing over here, this is a nerve cell in culture. Okay, I believe this is a primary neuron dissected out of a mouse. And you've got a pipette that's drawn up to the surface of it. And what they do is they suck on the pipette, create some negative pressure, a bit of a vacuum, and that blows a little hole between the pipette and the surface of the neuron. And what that allows you to do then is to monitor currents, ionic currents across the membrane of this neuron. And so if you look at wild type, okay, you see no currents whatsoever. But if you look at delta CR, okay, the form of the prion protein that's missing that central region, you see these very, very strong, what we call spontaneous currents. These are cationic currents. So ions are flowing readily across the surface of this neuron. What this ultimately does, and I should just mention, well, you know, how do we get these neurons? Well, this is dissected. This is a neuron dissected out of a healthy animal. This is a neuron dissected out of a transgenic animal that has that central region missing. Okay, so this seems to go one-to-one, okay, with the idea of the toxicity of the protein. And so sometimes we show this in terms of just the so-called spontaneous currents, and other times we show it as a histogram. We show both so you can see the strength of the data. What you're seeing down here are mutations that correspond to human prion diseases. So individuals, I can't quite read it, P101L, I believe, okay? Individuals, families that carry that polymorphism, that mutation, have a very, very high likelihood of developing spontaneous prion disease, genetic prion disease. Similarly with this mutation and that mutation down there. And almost hand-to-hand, or one-to-one, any kind of mutation that exists in families that have a high probability of developing familial prion disease will also, when introduced into a transgenic animal and expressed as a neuron, or transfected into simple cell models like N2A cells, HEX cells, and so forth, will generate these spontaneous currents. So this now provides us a really, really nice readout. So when we want to understand the toxicity, we don't have to go to a transgenic animal. We can just produce the protein. We can transfect it into an N2A cell, which is a neuroblastoma line, or into HEX cells, human embryonic kidney cells, and we can look for spontaneous currents. And that's basically our working mode now. So, right. So now it gets even more interesting. You can also trigger prion disease with antibodies, and or let's say a prion disease phenotype with antibodies. And I'm just going to mention a couple of them. And again, this was initially discovered both by the Harris lab as well as the Aguzzi lab, but the one that's most well known is called POM1. All right, and this is really, really interesting. If you take cells or brain slices or animals and inject the antibody directly into the central nervous system, okay, this POM1 antibody has an epitope on the C-terminal domain, and that will produce spontaneous currents. It will also produce death, when injected into healthy animals. On the other hand, POM11, which binds the N-terminal domain, does not produce toxicity. That one's absolutely fine, okay? And then finally, I'll just mention, I don't show the data here, but if you take the prion protein, if you express it not as the full-length protein, but just as the C-terminal domain, okay, and introduce POM1, it's not toxic, okay? So you see the riddle here? So there's something very strange going on. So an antibody that binds here is causing toxicity, but only when this region is present, not the region that carries the epitope for the antibody. So there's something very curious going on. And this is just showing, this is some fluorescent data showing the degradation arising from POM1 from various neurons grown in culture. Okay, so the way we now think of this is the protein has two domains. And so clearly that N-terminal domain is critical for toxicity. So we refer to that as the N-terminal toxic effector domain. And this is a language that's now commonly used in the prion field. And we have the C-terminal regulatory domain. So this domain somehow is regulating that domain. And this is where our work comes in, because we do the structural work, we bring in magnetic resonance, we're trying to understand what the heck is going on. And from a protein chemistry perspective, you would look at this and you would think there's no way this thing over here can be regulating that thing when that thing's just a bunch of disordered polypeptides sticking out in solution. So there's something really, really curious going on here. So what is that? So my prion colleagues here have covered this really, really nicely this morning. But as we now know, and I was mentioning earlier, the prion protein binds metal ions predominantly in the octorepeat domain, but also in a couple of non-octorepeat sites. It will bind copper, it'll bind zinc, those two physiological metals. It won't really bind any other metal ions, primarily those two. The chemistry is complex, so I won't go into a lot of detail. I'm going to mainly focus on this form right here. But I do want to mention, just like I believe Jan went through this in some detail, it can transition, and Caroline I think also as well, transition between these different occupancy modes. So the prion protein is responsive to the amount of copper it sees. And for those of you that are not familiar with this, copper, copper, iron, zinc, okay, the brain is rich with all of those metal ions, extracellular, intracellular, absolutely critical for physiological function. Right, and so there's also binding, so this is the octorepeat out here. There's these two non-octorepeat sites, as we refer to them, and I'll just mention that Liliana's lab has done an awful lot of work trying to understand these really important sites over here. And this is the paper that I was mentioning. Liliana and I wrote a review before we actually met each other in person And so we had a lot of fun working on this, and we had some Zooms and so forth, but that was a really great project. And I'll just mention we have a little structure right here of what each one of these sites look like, and I can talk about that later if anybody's interested. But to proof of concept, this was work now fairly old by Jake Pushy when he was in Canada, and he showed that the distribution of these essential metal ions, so this is a metal ion fluorescence experiment that's spatially resolved. And so what he did is he took different transgenic animals as well as wild type, dissected out their brain slices, these are coronal slices over here, and he looked at the distribution of metal ions as a function of prion protein expression. So I believe, okay, now I have to get close again. I think that's wild type, that's the knockout, and that's an overexpressor, okay? And you can see there's a really profound change, a profound, correspondence. And when you overexpress the prion protein, you also accumulate a lot of metal ions in the brain. So clearly, there's a causal link between these two. So I'm going to focus mainly on this form right here. And I'm going to talk about two different magnetic resonance techniques. And I went through some of this yesterday as part of our workshop. And so apologies for going through it again, but I'll explain very quickly how these things work. But we use nuclear magnetic resonance and electron paramagnetic resonance. And you'll see how both of these techniques apply. Now, let's see, right. So what we want to do is we want to answer this question, all right? So now, instead of thinking of the protein as being an ordered domain and a disordered domain completely separate, we now want to be a little bit more informed about this and say, wait, the prion protein binds metal ions, so let's put the metal ions in there, okay, in particular copper, and let's see if that creates some organization that helps us understand this regulatory interaction. And we call this, well, I'll refer to it in a couple of minutes, but a cis-regulatory interaction. So nuclear magnetic resonance, for those of you that are not familiar with it, in some ways it's very, very easy to do. It has a fancy name, HSQC, heteronuclear single quantum coherence spectroscopy of nuclear magnetic resonance. And you say that 10 times, you get very confused. What's really useful for us is that if you take a protein, so what you do is you express a protein with uniform N15 labeling. The standard isotope for nitrogen is N14. You express with N15. You can do this with enriched N15 ammonia. It's very, very, very common nowadays. Then what happens is you get a cross peak that's unique for each amino acid in your protein. And this is a really, really powerful tool for looking at drug-protein interactions, protein-protein interactions, metal-ion protein interactions, you name it, okay? And so what we're doing here is we're taking advantage of an effect called the paramagnetic relaxation enhancement effect, or PRE. Now what happens, copper 2 is paramagnetic. It has one unpaired electron. And if you can imagine what's happening in that electron sitting in a d orbital, it's flipping, that electron flips back and forth between its spin states, if you think back to freshman chemistry. And in doing so, that creates a fluctuating field nearby any amino acids that are close to that copper. And the fluctuating field broadens the HSQC lines. So the real simple readout is you have a black, so the black spectrum is in the absence of copper and the red spectrum is in the presence. And you can see that some amino acids are just fine. And I should mention, there's 200 peaks that show up, in this protein. This is just a small section to illustrate the point. And then other peaks in the presence of copper are eliminated. Okay, and that means that amino acid, that amino acid, that amino acid, et cetera, are close to where the copper is. These signals are not from the N-terminal domain. They're from the C-terminal domain, the regulatory domain. And if we map the amino acids that are affected by the presence of copper onto the regulatory C-terminal domain, we find that they all cluster on this surface right here. Okay. So it's all coming together on this one surface, and it's much clearer on this surface map representation that this patch is affected by the copper. And again, at this level, the copper is not binding directly to the C-terminal domain, it's binding to the N-terminal domain, and therefore this N-terminal domain must be coming over, carrying the copper with it, and I'll show you a rendering, and interacting with this surface right here. Okay, so now, electron paramagnetic resonance. So this is called Deer spectroscopy, double electron-electron resonance, and it goes along with a fairly fancy pulse sequence, and we don't have to go into the details. But the point here is that this is a sort of an orthogonal technique where we can look at the distance between paramagnetic centers. So it can be between a free radical and a metal ion, between metal ions, what have you. And one of the tricks that those of us in the EPR field do is that we introduce this stable paramagnetic side chain called a nitroxide, and we can introduce this anywhere we want into a protein with mutagenesis. It's very, very straightforward. And so what we wanted to do is ask the question, if we introduce a nitroxide or several nitroxides into the C-terminal domain, and then use DEER to measure the distance between the copper and those nitroxides, can we help localize where the copper is, okay? And so this is just a proof of concept of experiment where we generated a simple helical peptide. We put the nitroxide over here, we put the copper over here. We can do a molecular mechanics calculation to figure out what the average distance should be. These are what the data look like. It's called a dipolar evolution. And then if we take this and we apply a type of transform, it's similar to a Fourier transform, that gives us the blue line, and you can see there's really nice agreement between the model and the experiment. Okay, so now what we did is you need 4 distances to localize something in space. It's kind of like GPS, okay, but which uses 3 distances, and I could explain why if anybody's interested, but for protein we need 4. So we put a spin label here, okay, so, oh, I should mention also, the Prion protein has a disulfide bond right there. And so we use a non-coded unnatural amino acid introduced into the protein that allows us to spin label without interfering with any of the disulfide chemistry that's required for proper folding of the protein. This uses you know, a repurposed gene to be able to introduce this unusual amino acid, this unnatural amino acid, which is basically a phenylalanine with an acetyl group right there. And then we do a little bit of chemistry on that, and now we have this paramagnet introduced into the protein. So this side chain, in one copy of the protein is here, and another copy of the protein is here, another copy there, another copy there. So now we can generate 4 distances to the copper ion. And what we get is distance distribution. So these are the raw data, these are the transforms over here, and we get a distance distribution, and it's fairly broad. Either one of these really helps localize the copper, but what we have to do is find a distance or a localization for the copper that satisfies all of these distances simultaneously. And there we go. And so you can think of this as drawing a sphere around each one of the nitroxides in each copy of the protein where we did this. And it turns out that they all overlap in this region right here, and it's shown by this ellipsoid. So once again, this is the full-length protein with a single equivalent of copper. And now what we're finding is based on these distance distributions, these distance restraints, that the copper is localizing right here as represented by this ellipsoid. And you can turn it around and see it on the 2 different sides. And this is just a little movie demonstrating that. And again, I emphasize, it's not in this rendering binding solely to the C-terminal domain, it's binding to the N-terminal domain, and that's coming over and interacting with the C-terminal domain. So what this now means is we are now seeing the first evidence of what this regulatory interaction between the toxic N-terminal effector and the C-terminal regulatory domain is coming from, okay? And it's mediated by copper. So when copper binds to the N-terminal domain, it gathers up all that polypeptide, and that interacts with the C-terminal domain over here. And I'll show you data making it clear that this is in fact regulating the toxicity. We call this the cis interdomain interaction, okay? Cis meaning it's coming from 2 domains within the same protein, and those of us in the field, we just call it the cis interaction. So anyhow, so this is what we think is going on. We think this is how the N-terminal domain is being regulated. So a few things to point out. One is that here's the ellipsoid from the EPR data. The red are the PRE effects from the N-terminal data, or the NMR data, and you can see there's really great agreement between the 2, okay? So you have 2 different techniques both telling us the same thing, okay? That this is the important surface. In addition, everybody in this room, all of our prion proteins have a very highly conserved, negatively charged patch, okay, on this surface of the protein, this electrostatic patch. All mammals have this, sheep, goats, deer, cows, etc. And this has been a real curiosity. Why are these acidic amino acids so highly conserved forming this patch? Well, copper is positively charged. This is negatively charged, so perhaps it's facilitating this interaction, and we believe it is. And then finally, I mentioned the POM1 epitope, or the POM1 toxic antibody, right? Well, where's the epitope? Well, the epitope is right there, and it's literally right under where the copper N-terminal octorepeat domain would otherwise be. So now this generates a scenario that makes perfect sense. So what's happening is when you introduce, so the C-terminal domain regulates the otherwise toxic N-terminal domain, which if it's not regulated will punch holes into cells as measured by electrophysiology. And when you introduce this toxic antibody, even though it's binding over here, it's interfering with this domain-domain interaction and liberating the N-terminus, so then it can now interact with membranes in a toxic way. So now we have a scenario that we can use. And this is just showing the patch, the negative patch among the different kinds of species, rodents, you know, barnyard animals, you name it. Okay, so now what about delta CR? So proof of concept. So if we're right about this, okay, we should see an effect in delta CR, okay, the form of the protein that's artificially missing those middle 20 amino acids. And in fact, that's exactly what happens, okay? When you're missing that linker region between the copper carrying octarepeat and the C-terminal regulatory domain, we see we see many fewer amino acids affected by the paramagnetic, by the PRE. Okay. So now we have a scenario where we can understand this. Now, let me take it a step further. So this is more electrophysiology data. So this is delta CR, and if it's untreated, okay, we see the spontaneous currents that I was mentioning before. If we dose in copper, okay, and you have to be careful with copper, as those of us who work with it know, because it can aggregate on its own. So if we introduce copper as a pentaglycine complex, which allows the copper to be soluble and transfer to any other copper binding protein that has a reasonable affinity, all of a sudden it arrests those currents. So adding in copper reduces the currents. So that's also consistent with copper-mediated interdomain cis interaction, and it's shown in this histogram over here. If we take another version of the protein, and transfect into cells where it has delta CR, but it's also missing this segment right here that binds copper, okay, we see spontaneous currents, but adding in copper doesn't rescue. Okay, so that octarepeat domain is absolutely essential for that domain-domain interaction, and that's what quiets the currents and eliminates the toxicity. So we now have multiple forms of evidence showing that this domain-domain interaction is very important. Okay, so this is now another rendering showing the same thing. So the N-terminus of this is really important over here. This is the polybasic segment, and it's actually fairly well known in membrane physiology, biophysics, however you want to think of it. And it's a little bit counterintuitive, but it's very clear that cationic peptides will penetrate membranes. And so there are actually certain antibiotic peptides that behave this way. So they will interact with membranes, punch holes, create leakage, and this is probably the source of the toxicity that we're seeing. So it's all gathered up here. The polypeptide is gathered up by copper. The copper is really small. It's right in there, but this is forming this domain-domain interaction. Now, this is somewhat newer data over the last couple of years, but if we look at this face on and we strip away the N-terminal domain, we see that there are two C-terminal histidines. And for those of you, I should have mentioned this earlier, histidine, wherever you see histidine, you can have a good sense that there might be a metal ion binding, because this is the amino acid that proteins often use, okay, to coordinate different types of amino acids. And of course, it's the as I showed you earlier, it's the histidines that are coming around the copper ion. So we wondered whether these C-terminal histidines were somehow playing a role in this regulatory interaction, because they're very close where the copper octarepeat is nestling in. So once again, we wanted to do an experiment and look at this. And so once again, you see the octarepeat domain folded around the copper. There's the copper. This is a bond line diagram showing that with histidines around the copper. And so we want to query whether copper binding to the octarepeat domain, even though there's enough histidines around it, okay, to fully coordinate it, if somehow these histidines are also playing a role in this regulatory interaction. So we just did a really simple experiment. And what we did is we knocked out the C-terminal histidines and we replaced them with tyrosine, which is isosteric. And sure enough, we see this is the PRE data. We see a very different interaction. Instead of resonances being broadened by the copper in the vicinity of the two histidines, as we had seen previously, now that's gone, but there's still some interaction over here, and this is more localized over that electrostatic patch that I was mentioning before. So those C-terminal histidines are playing a significant role in this domain-domain interaction. So now we want to know, so we're chemists, and we want to know the details, right? We want to know what's really going on. So how the heck is a fifth, if this is the case, how is a fifth histidine somehow coordinating to this copper ion? And you can imagine there's a couple of different ways. One is that it could come in axially, okay, and form a bond this way, or one of these could be dissociating, okay, and this could be coming in what we call a histidine swap, where this is a C-terminal histidine coming in and coordinating the copper. So we generated two forms of the protein. I've showed you both of these now. This is wild type, and we call this DM double mutant, where we've eliminated the two C-terminal histidines. And these are just EPR spectra. And for those of you that are comfortable with EPR spectra, this looks really, really familiar. I won't go into the details, but the main point here is that without going into any EPR theory whatsoever, if we look at the protein, the double mutant, and the wild type, and we compare the normal with N14, N15, simulations, et cetera, they all superimpose on top of each other. So there's nothing changing in the equatorial environment around the copper. We can also do, and this is where Dave Britt's work comes in and Lizzie Tao's work, do ENDOR. And this is a sort of combined EPR NMR experiment where you're using an EPR readout, but you're stimulating the nuclei by turning on radio frequency, extended radio frequency pulses. And so you can look for specific frequencies. And once again, I'll just say we see no difference between the double mutant and the wild type. So this suggests once again that the environment is staying exactly the same. So what the heck is going on? So it kind of suggests that one of these equatorial histidines is being replaced by a C-terminal histidine. But if that's the case, we want to really nail this down and show that there's something, perhaps water, okay, coming along this axial site. So we did an experiment. So water, O16 water has no magnetic resonance properties in terms of the oxygen whatsoever. So we purchased O17 water, and I like to point out that this costs, I think it's $2 million per liter. So we bought half of a milliliter, and you know, which is barely affordable in and of itself. And we did some ENDOR experiments, pulse ENDOR experiments again with the Brit Lab, and we see the O17 coupling. And this is a very fancy EPR experiment called HiSCORE, hyperfine sublevel correlation spectroscopy. And in the presence of O17 water, we see these couplings, which are equivalent to those couplings. So we now know that what's going on is that the C-terminal histidines are participating in this domain-domain regulatory interaction by replacing one of the octarepeat histidines. And so we have a model that looks like this now. So this histidine, this one, and this one are coordinating around the copper, but this one is from the C-terminal domain. Okay, now, and I'll just say for the magnetic resonance folks in the audience, we now have all the couplings worked out for all the nitrogens, protons, O17, et cetera. Okay, now, this is unpublished, but we're really excited about this. What you'd really like to do is have different isotopic labelings for C-terminal histidines versus N-terminal histidines. And there are certain magnetic resonance techniques that I'll show you in a second that allow you to distinguish N14 histidine coupling to N15 histidine coupling. So what we wanted to do was express the protein where we had an N14 C-terminal domain and an N15 N-terminal domain. And this requires transpeptidation, ligation chemistry. And as I pointed out yesterday when we were talking about this, this took us 2 years to get this to work. But a graduate student in my lab finally has this working. We use an enzyme called sortase A, and this is basically a proteolytic enzyme, carries out proteolysis, and you run it in reverse, and you know, an enzyme that's a protease, you run it in reverse, it becomes a ligase. So this can ligate two polypeptide segments together. So we finally got this to work. We now have prion protein that's selectively, so-called segmentally labeled, where we have N15 in the N-terminus and N14 in the C-terminus. And we've gone on and characterized this, and I'll just jump right to the results. And that is, so this is what's called an E-seam spectrum, and this is yet another way of using EPR to read out nuclear frequencies, nuclear couplings. And so we'll just do this by fingerprint, okay, without really interpreting any of the lines. So this is what you have when you have histidine that's N15, okay? This is what you have when you have histidine that's N14, and this is our segmentally labeled protein, and you see that we now see the signals from both of these superimposed on top of each other. So the bottom line is that we are definitely seeing now a positive result, not a process by elimination, but a positive result showing that the C-terminal histidine is actually playing a role in coordinating directly to copper. Okay, so this helps us solidify this model. So now we think this is on very, very good standing from a biophysical perspective, from an electrophysiological perspective, and so forth, that the C-terminal domain regulates the N-terminal domain through mutual copper coordination. All right, now let's see. Yeah, so that's just showing what I showed you before. All right, Okay, so back to proof of concept. Well, if that's the case, then that means those C-terminal histidines should be important in terms of the prion protein's toxicity. So we go back to the electrophysiology data with David Harris, and we look at spontaneous currents from wild-type protein and our double mutant. And this was now just the protein, showing over here, where the two C-terminal histidines have been replaced by tyrosine, and it is very, very toxic. It produces spontaneous currents. So we know not only see a domain-domain interaction and a positive, and we now can characterize the domain-domain interaction, we now have a physiological readout that in fact that interaction is cytotoxic. Okay, so this will be the last part that I'll go through, and hopefully I'm not running over by too much. So the carbohydrates, the glycans, what are these guys doing? So those of us in the protein magnetic resonance world, we always express our proteins in E. coli. E. coli does not glycosylate proteins. So we're always working with naked proteins, and we think we know what's going on, okay? And sometimes we don't know what's going on because we don't have a protein that really reflects what's happening in vivo. And so once we identified that this regulatory surface over here, we started becoming a little nervous about what these glycans, what these carbohydrates might be doing. Because you can imagine, you just look at this and you think, oh my gosh, well, if copper binds here, yeah, sure, in the absence of these, you can imagine this copper bundles up this and comes over and interacts with that surface. But this looks like it would sterically block that from happening, okay? So we worried about this. We figured we have to test this directly. We have to know what's going on. Well, there's a problem. And the problem is that when you express a protein with N15, so you can do nuclear magnetic resonance experiments, HSQC experiments, you're doing it in E. coli, so it's always unglycosylated. So we had to figure out a way to glycosylate an N15 expressed protein. So one of my former students, Kevin Schilling, and I'll point out right now, this is my only organic chemistry paper ever in my entire life. and will probably remain the only one in my entire life. But my student Kevin figured out a way to do this using chemistry that Peter Schultz had developed. And basically what he did was we once again used the uncoded amino acid, unnatural amino acid. We introduced paracetyl phenylalanines in the locations where the carbohydrates would otherwise be. And these were asparagine-linked carbohydrates, I should mention. And we have some standard chemistry that allows us to generate a monosaccharide reagent that attaches right to those, and that creates this step over here. And then we use glycotransferases to elongate those in a step-by-step fashion. So it's not a full carbohydrate, but it's a trisaccharide that's a reasonable mimetic, okay, for looking at this. And so now we have a model for a glycosylated N15 labeled prion protein. And so we now look at the consequences of this. So this is showing, once again, the magnetic resonance data and the paramagnetic relaxation enhancement through the NMR measurements, okay, as it affects the various amino acids. So the data just very qualitatively are shown here. So this is wild type, unglycosylated. This is what I showed you before, okay? So this is the experiment that now made us nervous. So this is wild type, but now with artificial glycans introduced. And we don't lose the paramagnetic relaxation enhancement. It actually shifts and prefers to interact with this helix right here, okay? And this is helix number 2 of the C-terminal domain. And in fact, it nestles right up to the linkage point of where that carbohydrate, where that glycan is adding, okay? Even further, if we take unglycosylated and replace the histidines with tyrosines, we still get an interaction. And then if we, let's see, replace, right, if we glycosylate, okay, and replace the C-terminal histidines, we get an interaction, okay, that looks almost as strong as the interaction we're seeing up here. So in other words, yeah, the histidines are part of the interaction, but it almost looks like, counter to what we were thinking, that the glycans might even be more important in facilitating this interaction in vivo. And I should mention when David Harris says the electrophysiology, that's a normal expressed prion protein that has its glycans. Okay, so now with that, we go back to the electrophysiology. And this is what we found really shocking. And we just published this a couple of months ago. Okay, now I have to get close to be able to read this. All right, so this is delta CR. So in the taxonomy of all the different forms of the prion protein, the one that produces the strongest spontaneous currents has always been delta CR at -70 millivolts resting potential. This is now our deglycosylated. Okay, so he's now expressing, he's now knocked out the two asparagines that would otherwise link to the glycans. And when that's expressed in, I think this is in N2A cells, I believe. Yes, N2A cells, the neuroblastoma line. We get spontaneous currents that are almost as strong as delta CR, okay? And we see it at two different resting potentials. So basically, by eliminating the glycans, we produce a toxic form of the prion protein that rivals the most toxic form previously seen, even more toxic than the familial mutants that are introduced. It pretty much parallels delta CR, and there's some other data in there. But so now what we're seeing is that the glycans, as measured through the magnetic resonance data, are not only enhancing this domain-domain interaction, but also from the electrophysiology, they're also modulating and tamping down the toxicity that would otherwise arise from an unregulated N-terminal domain. So this is now kind of a simplistic schematic of how we think about things. So this is the wild-type protein over here. So we have copper facilitating this domain-domain interaction. You can barely see it, but there's negatively charged amino acids over there, and then we have the glycans. And we think all of these are facilitating this incredibly important interaction that regulates the protein's toxicity. If you knock out the C-terminal histidines, you weaken the interaction. Delta CR eliminates part of the linkage, the linker domain that also enhances that. And if you eliminate the glycans, likewise. So these forms, this form is mildly toxic, but these two forms parallel each other in terms of toxic, and they are the most toxic monomeric forms of the prion protein. So we feel at this point, and this is just summing up, saying basically we think there's three complementary interactions: his coordination, acidic residues, and glycans, and they all work together. And given how toxic the N-terminal domain is when it's not regulated, it's nice that there's some redundancy if we want to think of it in those forms. So there's a lot of things we still don't know, and most things we still don't know. How do the glycans stabilize this? We really don't have any real structural data at this point. What's the role in prion disease, and likewise in Alzheimer's disease? And so what we now think, and this is again just sort of a schematic, is as in the previous session, Vic, Jan, Carolina were talking about the modulation of various receptors. So what is the function of the prion protein? It is thought to be a protein that modulates the function of other proteins, transmembrane proteins, and glutamate receptors, the NMDA, AMPA receptor, are targets that have come up, okay, in the physiology literature. And so very schematically, here's the C-terminal domain of the prion protein. We have copper binding to the N-terminal domain that introduces this domain-domain interaction, and this positions the N-terminal domain to modulate the behavior of whatever receptor it happens to be nearby, and delta CR weakens this interaction and either causes a different type of signaling through the receptor or introduces toxicity through damaging transmembrane currents. And one can even speculate and go further than that. We were talking earlier about the interaction of A-beta. So A-beta aggregates may be interacting with this interaction, causing downstream effects, intracellular alterations in intracellular signaling, dysfunction of transmembrane receptors, and so forth. So there's a lot we still don't know, but at least I feel like we're narrowing in on what is going on with the prion protein in terms of the domain-domain interaction, the cis interaction, how it's regulated by copper, its primary high-affinity metal ion, and how this regulatory interaction that has been a curiosity in the prion field for so many years, how this regulation is taking place. So we have much to learn as far as this goes. But as we were talking about earlier, Alzheimer's disease is much, much more prevalent, and it is now known, and this is something I'd be happy to talk about, in the Q&A or later one-on-one. It is now known that the prion protein is actually one of the primary receptors for A-beta aggregates. And in fact, a lot of the deleterious physiology that one observes in transgenic animals and laboratory mice that's introduced by A-beta aggregates, if you use those same A-beta aggregates and you look at a prion animal, a transgenic animal that's lacking the prion protein, the A-beta aggregates don't have nearly the same level of toxicity. So the prion protein, not just being a protein of prion disease, could ultimately end up playing a major role in terms of our understanding of the most prevalent disease being Alzheimer's disease. So let me, I think that's it. Yeah, so this is the lab, and there's Jan and Vic. They were visiting for a couple of weeks, and I keep telling them they didn't finish their work and they have to come back for more. and that extends to anybody else in Liliana's lab. And there's Fran and Amanda and Celia and Kevin, and Jack's our NMR facility manager, and it's a wonderful group. And let me just mention down here, we're funded by the National Institutes of Health, and we're super grateful for this Alianza grant that not only funds some of this work, but also brought us all together for this incredible workshop. And yeah, and there's all the collaborators again, and I will finish there. Thank you.
We have time for questions.
I understood that.
Thank you very much for your talk. It's amazing how profoundly you get to know the proteins with all this chemistry. But I still have the question, how do you contract the disease?
Okay, very good question. So this could be a long answer, but I'll try to get to the point. So first of all, in individuals that have prion disease, one thinks that they always have prion aggregates, okay? And there are many cases where they don't. They do the postmortem histology, and they find that there's a prevalence of prion protein, in a certain region, but not necessarily as beta sheet-rich aggregates, okay? So how do you get the disease? Well, first of all, there's spontaneous disease. Unlucky individuals the prion protein will misfold spontaneously, similar to Alzheimer's disease, similar to Parkinson's disease, and those aggregates then start growing. Okay, they seed an aggregation process in whatever region of the brain that takes place. There's also familial mutations with Alzheimer's disease. You have the ApoE poly, alleles that predispose people to getting it, and that changes the regulation of A beta. And then, of course, there's the infectious route, and that comes about. There have been a number of cases. So first of all, when tainted tissue is consumed, okay, and it goes down through the gut into the intestines, okay, those are heavily innervated areas, right? And so there are pathways from there straight back to the brain. And in fact, in Parkinson's disease, this has been known in the neurodegenerative field for years. But people that suffer from Parkinson's disease, not to be overly graphic, but one of their earliest symptoms is not the neurodegenerative phenotype, but is constipation. Their bowels stop working, okay? And this is now believed that, in fact, the aggregates start there and they propagate back up through the peripheral nerves back to the brain. So there's lots of pathways.
So you consume, like, I was in England when the mad cow disease started. Yes, I was doing
So you probably weren't allowed to donate blood.
Exactly.
Yeah. Yeah.
And for other reasons also, but that was the first one. When I was doing my master's, it was the thing of the mad cow disease. And the thing is that they said, well, you shouldn't eat like burgers or things.
Right.
You shouldn't eat meat that is contaminated. But I never really understood how you ingest some protein that is deficient, and then it propagates, or it makes your own prion proteins to unfold?
Yeah, well, right, okay, exactly. So the way I would think of it, and Liliana can set me straight on this, but I would think of it is that it already has the prion aggregates in the meat that that the individual will consume. That those aggregates then propagate and they start interacting. You then become the substrate producer, right? You're producing your own prion protein, and that adds on to those aggregates. And in fact, if you take, first of all, if you take scrapie tissue out of a diseased animal, okay, a transgenic, laboratory mouse, take scrapie tissue out, implant it into the brain of a healthy animal, it will grow. The scrapie material will grow, okay? Alternatively, take that same tissue, inject it into a PRP knockout, okay? Not only will you not see anything, right? It's not even toxic, okay?
Thank you very much.
Yeah, so just to add to that, it's as if the aggregate of the prion serves as a template, and it can add the monomers and grow the aggregate. So, questions?
Thank you. So, by what you presented, if the prion disease or the mutations of the protein itself cause these spontaneous currents, then you present that maybe the prion protein is regulating itself, the
Oh,
like NMDA and AMPA,
yeah.
and that, I mean, my question is that like the most likely scenario of what's happening if it's not about the aggregates?
Let me, it's a really good question. Let me paraphrase it, okay? So I think what you're asking is if there are familial mutations, and if our domain-domain interaction is important for regulating the prion protein, do the familial mutations impact that domain-domain interaction, right? They do, okay? I didn't show the data here, but that was actually one of our very first experiments, and we were actually surprised. And in fact, we went after the obvious ones that were on the C-terminal domain where we saw the interaction. And then we had a group meeting with Stan Prusner, who's the Nobel laureate, at UC San Francisco. And he goes, oh, you should look at P101L, or I think it's P102L in mouse, or vice versa. And that's in the linker region. And we didn't think we would see anything, but we did. That also interfered. So your question also leads to a somewhat broader question. What is the disposition of that linker? Okay, how is that thing structured between the two domains? Because there's a lot of polypeptide there, and we don't know. So this is one of the things with the segmental labeling that I showed you before. This is one of the things I hope we can start getting a handle on, because by having segments of the protein N15 labeling, that will clean up the NMR spectrum, and we can then start interrogating what's going on structurally with that linker region. But you're absolutely right. I mean, if our scenario is correct, then the familial mutations should also be interfering with the domain interaction, and they do.
Okay.
Yeah.
Thank you.
Thank you for your very nice talk. What's the function of the GPI anchor? Is just to have the protein in the membrane, in plasma membrane, but also in inner membrane?
Yeah, so I think of it as mainly an extracellular protein. It is modulating nearby receptors, so it has to be anchored to the protein. And so I think that's what its function is. It can't carry out its function in terms of modulating other membrane receptors unless it's localized to the membrane, and that's through the GPI anchor. But there's another component to this, and that is when I was mentioning before about introducing scraping material into animals that lack PRP, and how the tissue, the scraping material isn't toxic. There is a transgenic. It's called the anchorless prion protein, and this was done at the Hamilton lab in Montana, which is the NIH outpost for infectious diseases, and they produced prion protein. They produce a transgenic animal that lacks the code for the GPI anchor. And these animals produce lots and lots of extracellular PRP. And when you introduce scrapie, they generate lots and lots of aggregation with very little neurodegeneration. Okay, yeah. Well, I don't know about inflammatory interaction. That's a good question. Yeah. But they do. I mean, look. If you're going to have all these protein aggregates, at some point they're going to become toxic. They're starting to crowd out other functions. But in terms of that really acute toxicity associated with PRP, that is eliminated in the anchorless prion protein.
Hello, thanks for the conference.
Sure,
it's really nice. There are any experiments that show if the cleaved protein has any toxic effect on the cells?
Yeah, so it is, usually when we think about, cleaved protein, as you well know, PRP undergoes alpha cleavage and beta cleavage, and alpha cleaved is a normal proteolytic event for PRP, and that's yet a whole nother story, okay? That alpha cleavage is actually protective against aggregation, okay? And it can't be, once you cleave off the N-terminal domain, you're not going to generate spontaneous currents, so you eliminate that as well, all right? Beta cleavage, okay, and it's an interesting point, beta cleavage leaves a lot of polypeptide there that's fairly hydrophobic, and can actually enhance protein aggregation. So beta cleavage is something you don't want happening, okay, and alpha cleavage is something you do want happening. And there's parallels in Alzheimer's disease, right? Because if you have alpha and gamma, if you have beta and gamma, that's deleterious because that produces the full length A beta 1 to 40, 1 to 42, et cetera. But if you have alpha cleavage as well, that cuts that in half and eliminates the toxicity associated with A beta.
Okay, thanks.
Thank you for your talk. I have a very complex question.
Uh-oh.
Now that you know the mechanism of this kind of interactions and coordination with Cooper, what would be the pharmacological or therapeutic approach to treat this disease?
Oh, that's a great question.
Because because you mentioned that you can play with the toxic region and with the regulatory region, right?
So there is a couple, Eric and Sonia, who live in Boston. They work at Harvard. Eric, it was I think a computer programmer, and Sonia was an attorney. And her mother died. Sonia's mother died at a young age, and they realized it was prion disease, and it was a genetic form of prion disease. So Sonia was genotyped, and she found that she carries the same thing. So they are working harder than anybody in the world to try to find a cure for prion disease. Sonia's now in her 40s, and so her days are numbered. And I've actually, if you go to prion meetings, you meet people who carry the familial mutations, and they know that they won't live past the age of 50, 55, something like that. So there's many, many different approaches, and the standard one is, since the prion protein is the substrate, is to just knock down its expression. And this is done with various messenger RNA type of therapeutics. Unfortunately, at least in animal models, nothing has really worked yet. But a little bit different than your question, but something that interests Liliana and me and folks in her lab is with Alzheimer's disease. If Alzheimer's disease, if A beta is trafficked, okay, and enhanced and moved into the cell by the prion protein, which is a whole nother story, okay, and something we have data on. If that's really critical in Alzheimer's disease, and again, much more prevalent, could we think of therapeutics that would interfere with that domain-domain interaction? This is exactly what some of Liliana's folks are looking at. We're very interested in it as well. And so I think, yeah, if you can think of, and now it gets down to molecule, so you can start thinking about screening experiments, and so forth, to try to find molecules that will interfere with this. I worry a little bit, about anything too simplistic, but I think it basically involves screening and a lot of thinking. But yeah, it's a good point.
Any more questions? Well, I have a quick one, Glenn. So you showed that copper stabilizes the cis conformation, and that glycans also do. But if I observed well, the glycan experiment was with copper, right? So can glycans stabilize the cis conformation in the absence of copper? I guess you would have to put some. I don't know. I guess you will have to put some paramagnetic probe on the glycan to see. Yeah. No, that's okay. Now we have to do that. So just wondering. And also wondering what will be the mechanism, which you already said we don't know, and it's the perspective. But I was just thinking about the glycans and the copper. But we should be able to look at that by NMR even without the PRE, if we can get some decent assignments and figure out what's going on in the linker region and see some resolve residues from the N-terminal domain. We should be able to look at that. Just curious. Yeah. Thank you. Any more questions? The one.
Thank you, Glenn, for your nice talk. I have two small questions. The first is about the possibility that the cis interaction stabilize PRPC in the lipid drops. Thinking that PRPC is anchored through GPI, and GPI it's very flexible in the membrane, and actually PRP also can be endocytosed. And thus, I think this could be a way to explain how cis conformation is important to the interaction for the different types of glutamatergic receptors.
Yeah, I agree. I think it's a really good point. So what Jan is referring to is this is a very, very old experiment that's never been thoroughly explained, right? And this is, so PRP is cycled through endocytosis, and interestingly, most of it is cycled back to the plasma membrane by exocytosis. And copper and zinc both stimulate that process very aggressively. And so the question becomes, and also you can stimulate it with antibodies that cross-link. So it kind of suggests that when copper is there, or zinc, it's facilitating a domain-domain interaction between an intermolecular. And yeah, I think it's a reasonable hypothesis, but I don't have a model for it at this point, other than maybe shared histidines between copies of PRP. Yeah.
Yes. And the other question, it's about mutations that introduce extra octorepeat in the N-terminal region of PRP. I don't know if you test if these extra octorepeats can avoid the cis interaction.
Okay. We published on this, and we didn't know what we were doing about 15 years ago. Okay. We expressed, and you will appreciate this, we expressed PRP, but actually I think we work mainly with octorepeat models, okay, polypeptides, from 4 repeats, 5, 6, 7, 8, etc. And you know about the different binding modes. And what happens is the affinity for component 3 goes way up, okay? And the protein gets locked in that conformation. So I don't know how to answer your question. I mean, I could imagine that maybe part of its physiology is important to be able to transition, you know, between component 3 and component 1. But one could also imagine that you have an elongated N-terminal domain, you have less regulation of the N-terminal domain. It's longer, there's more polypeptide to regulate, and that polybasic N-terminus is now free to interact with membranes. But we haven't, you know, this was a paper by Stevens et al. And yeah, and we interpreted it back then without knowing anything about the cis interaction, you know, that we now know. And so we should go back and look at that. It's a good point. Yeah.
Thank you so much.
Sure, yeah.
All right, if there's no more questions, let's thank Glenn for a great seminar. Thank you. And thank you all for coming. And for those of you who are in the workshop, we adjourn here at 4 p.m. And even if you're not in the workshop, actually at 4 p.m., Glenn will give a short presentation on another project from his lab.
Oh, can you tell them what?
Yeah. So this is a totally different, for those of you in the workshop that want to hear about this. It's a really interesting story on it's not Prion disease, it's not neurodegeneration, it's a different type of disease called lysosomal storage. And the real quick introduction is our cells have a great protein recycling system. Basically, we express proteins, extracellular proteins, when we no longer need them, they're brought back into the cell for endocytosis. They go down the endolysosome pathway. They're broken down into constituent amino acids, and those constituent amino acids actually come out of the lysosome back to the cytosol so they can be produced into proteins. And when that process breaks down, you end up accumulating amino acids in the lysosome, and that is highly toxic. So we completed a study, in fact, published last year in Cell, with researchers at Stanford, where we used PCR technique along with their cryo-electron microscopy crystallography to solve the structure and look at the domain-domain dynamics of one of these transporting genes. It's a really cool story.
Yeah. So if you want to hear this story, Glenn will give this story this afternoon at 4 p.m. It will be short, if he says, maybe half an hour. And then we will continue with our EC spin workshops. So see you at 4.